Hello students, today our topic is Kingdom Fungi. This is a very important topic, and in this lecture we will discuss the general characters of Kingdom Fungi, and we'll also see how fungi are classified into different groups, into different taxonomic groups. Now let's begin. The word fungi was for the first time coined by Carolus Linnaeus in 1973. Now this fungi it constitutes a separate kingdom, as we already studied in case of five kingdom classification. This kingdom fungi, because of some peculiar characteristic features, it was placed in a separate kingdom. And there are about 100,000 species of fungi. It's a very diverse group of organisms, and new species of fungi they are being discovered every now and then. The study of fungi is known as mycology. The study of fungi is known as mycology, and anybody studying fungi or carrying out research on fungi is known as mycologist. Is known as mycologist. So the study of fungi is mycology, and anybody studying mycology is mycologist. Heinrich Anton de Barry, he was a German mycologist. He is known as the father of mycology. And Sir Edwin John Butler is considered as the father of Indian mycology. Now the fungi, they are eukaryotic. They possess all the characteristic features of eukaryotic cells. They are multicellular except for yeast. All fungi are multicellular except for yeast, which is unicellular. They are achlorophyllous. They do not possess chlorophyll, and as such they cannot carry out the process of photosynthesis. They are thalloid. Their body is not differentiated into roots, leaves and stem. They may have root like structures, they may have stem like structures, but they are not true roots or true leaves or true stem. They are spore bearing organisms. They do not possess seeds as in case of higher organisms. They have spores. The spores germinate under favorable conditions and give rise to the full-fledged organism. Now here I want to add one more point. They are also non-vascular. Non-vascular. They do not possess vascular tissues, xylem and phloem. They are heterotrophic. They obtain their nutrition from other organisms. Now they may be either saprotrophs. They obtain their nutrition from the decaying materials. The examples include rhizopes, aspergillus, penicillium, agaricus, and morchula. These are some of the examples of saprotrophic these fungi. I have included one photograph here. This is the Rhizopes stolonifer. It grows on the decaying bread and obtains its nutrition from the decaying bread. Now they may also be parasites. These parasites they obtain their nutrition from other organisms, from the hosts. Examples include Candida, Puccinia, Trichophyton. Here I have included one example. This is cordyceps. It's a fungus, this one, and it is a parasite on ants, and it obtains its nutrition by growing on these ants. Ultimately the ants they become dead, but these dead ants they still show some movements because of the growth of these fungi, cordyceps, and as such they look like those zombies. They are dead, but still they show some movement. Now from this slide, if we have to define what fungi actually is, what fungi actually are, and we can define them as eukaryotic, multicellular, achlorophyllous, thalloid, spore-bearing, heterotrophic organisms. So they are a group of eukaryotic, multicellular, achlorophyllous, thalloid, spore-bearing, heterotrophic organisms, and they constitute a separate kingdom. Now, what are these fungi made up of? Actually, the fungi, they consist of cylinder thread-like structures. Cylinder thread-like structures. Here I will show you this one, this part. It is a cylinder thread-like structure. It is known as hyphae. Now, if we have a network of hyphae, it's known as mycelium. Since this part which I have this encircled, this one, it is the hyphae. Now if we have the network of hyphae, it is mycelium. So the whole structure is mycelium. This part is the hyphae. Now the mycelium may be septate if we have these septa. These are known as septa. If we have septa, each cell is a separate cell. We have these wall-like structures. So such a mycelium is known as septate. When these septa are absent, the nuclei are present in the same cytoplasm. All the nuclei are present within the same cytoplasm. Then such type of mycelium is known as synostic mycelium. Now fruiting body is the spore producing organ of fungus. In case of fungus, we have mycelium. Actually, this is the structure of a typical fungus. Now there are two parts. One is the fruiting body and another is the mycelium. Now actually this whole fungus is made up of mycelium. Mycelium is here also, but this part, it is concerned with the production of the spores, and it's known as fruiting body. And when we study the reproduction in fungi, we'll study this structure in whole, and this part is mycelium. The cell wall in case of fungi is made up of chitin. It is a polymer of carbohydrates, or it is made up of cellulose. You should note here that this cellulose is somewhat different than that of the cellulose found in case of the higher plants. This is actually fungal cellulose. Should write it as fungal cellulose. And polysaccharides are also present in the cell wall. Now the reserve food material is glycogen. It is a branched polysaccharide of glucose. It is the reserve food material of fungi. Now let's move ahead. Now we move to classification of fungi. The classification in case of fungi is based on the structure of mycelium or the type of sexual spore. First, we look at the classification based on the structure of mycelium. Now, based on the structure of mycelium, we have two types of fungi. One is aseptate fungi. These fungi, they do not have septa in the mycelium. Such fungi, they are also known as cynostic fungi, as we studied in the earlier slide. And we have septate fungi. These fungi, they have the cross walls, they have the septa. So based on the mycelium, we have two types of fungi. We have aseptate fungi and we have septate fungi. Now these septate fungi, they are of three types. Septate fungi are of three types based on the structure of the septa. Now based on the structure of the septa, they are of three types. Now first First category includes those fungi which have septa and they have a simple pore within the septa for the cell to cell movement of the materials, cell to cell movement of the material. So in first category septa are present and there is a simple pore present in each septa for cell to cell movement of the materials. Now second category includes the septate fungi which have septa and they also have a pore, but the pore is barrel shaped. It's a this pore is just like a barrel shaped swelling and it is known as dolipore septa or dolipore septum. Now third category is in these cases, in these mycelium we have septa. Septa is present, but we do not have any pore, or very little information is available about this group of fungi, this group of septate fungi. In some cases you'll find one nuclei, in others you'll find two nuclei. Whether the pores are generally absent or they may be present, but very little information is available about this group. So we have three groups based on the septa in case of the septate fungi. So septate fungi, they are categorized into three groups. We have fungi which have a simple pore in their septum. We have fungi who have the dolipore septum, and third category of fungi in which the septal pore may be absent. Now this all this classification is based on the structure of the mycelium. Now all the fungi which are cenocytic, which are aseptate, they were included in the group Phycomycetes. So all the aseptate fungi, they are included into the group Phycomycetes, and all the fungi which possess the simple pore in their septa, they are included into Ascomycetes, and those which possess dolipore septum, they are included into Basidiomycetes. Now all those fungi which either have septa and the septal pore is absent, or very little information is available about the type of septal pore or the type of septa, all these fungi, they are included in the group Deuteriomycetes. Now based on the structure of mycelium, we have now four categories of fungi, four groups of fungi. We have these Phycomycetes, we have Ascomycetes, we have Basidiomycetes and Deuteriomycetes. Now this Deuteriomycetes, it's also known as Fungi imperfecti. We'll get to know why actually it is known as Fungi imperfecti as we move. Now this classification is all based on the structure of mycelium. Now a scientist called Alexopoulos, he's a very famous scientist. He has contributed a lot in the classification of these fungi. He said that this classification is based on mycelium. It is based on mycelium structure. So this classification is not reliable. Now he said that the classification should basically actually be based on the type of sexual spore. So he said that classification of the fungi, it should be based on the type of sexual spore they produce. Now he said that instead of these Phycomycetes, he included two groups of fungi, zygomycetes and oomycetes. He said all the fungi that produce zygospore, they will be included into zygomycetes, and those fungi which produce oospore will be included into oospores. Those which produce ascospore will be included into ascomycetes. Similarly, those fungi which produce basidiospore, they will be included into basidiomycetes. All these spores, zygospore, oospore, ascospore and basidiospore, they are the products of sexual reproduction. During sexual reproduction, all these fungi, they produce certain spores and they are named differently. The spores which are produced by zygomycete, they are known as zygospores. So this classification is completely based on the type of spore that's produced during sexual reproduction. Now, in case of deuteromycetes, the sexual reproduction is absent. So we have 1, 2, 3, 4, and 5 categories. We have category 1, that's zygomycetes. It includes all the fungi which produce zygospore. Oomycete includes all the fungi which produce oospore. Ascomycetes includes ascospore producing fungi, and basidiomycetes includes basidiospore producing fungi. And in case of deuteromycetes, the sexual reproduction is not known. The information about the sexual reproduction is not available. As such, they are known as fungi imperfecti. Now, this fungi imperfecti, it includes a heterogeneous. It's a heterogeneous group of fungi. For example, if for example we have a fungus A, it is included in the deuteromycetes because it is sexual stages of reproduction. They are not known. It is a sexual spore is not known, so it's included into deuteromycetes. Now somebody carries research on this fungus and he discovered that actually this A, it performs sexual reproduction, and he discovers that this fungus A, it produces ascospore. It produces ascospore. So as he discovers that this A, it produces ascospore, it will be immediately removed from this category, from this group deuteromycetes, and included into ascomycetes, because ascomycetes includes all the fungi which produce ascospore. Or somebody discovers that this A, it produces basidiospore, it will be immediately removed from deuteromycetes and included into basidiomycetes, because basidiomycetes includes all the fungi which actually produce basidiospores. So this reproduction or this classification is based off Alex Paulus. It is based on the type of sexual spore that is produced within the fungi. Now, if we look at the asexual spores that are produced in these different categories of fungi, different groups of fungi, we have zoospore and aplanospore. They are produced in case of zygomycetes and oomycetes. In case of zygomycetes and oomycetes, we have zoospore and aplanospore. Zoospore is motile, while as aplanospore is non-motile. In case of ascomycetes, the asexual spore is conidia. In case of basidiomycetes, asexual reproduction is absent. Then in case of deuteromycetes, we have conidia. Now, as we move from lower fungi to higher fungi, we'll see that higher fungi, they do not have asexual, they do not perform asexual reproduction. They generally reproduce via sexual reproduction, because if you see here, zygomycete, it reproduces asexually, but it produces both zoospore and aplanospore is produced in these categories. In case of ascomycetes, you have only conidia, and in case of basidiospores, asexual reproduction is generally absent, and if it is present in some members, then they will produce conidia. So as we grow from lower fungi to higher fungi, the ability of these fungi to reproduce asexually actually decreases. Now you must be wondering what actually these zygospore, oospore, ascospore, aplanospore, conidia, what actually they are. Actually, these are the products of reproduction. Zygospore, oospore, ascospore, and basidiospore, they are produced during the sexual reproduction. They are the products of sexual reproduction. Zoospore, aplanospore, and conidia, they are the products of asexual reproduction. And how they develop, what they look like, and their structure and all that, this all will be discussed in our next lecture, when we'll study each of the groups separately, when we study all the each of the groups, zygomycetes, oomycetes, ascomycetes, basidiomycetes, and deuteromycetes separately. And when we'll study the reproduction in fungi, we'll discuss all these spores. So here we have used them for the sake of the classification. We have to study them in detail in the next lecture. Thank you very much. Have a nice day.